it is my pleasure to now introduce our speaker, Laura. Laura Wiggins is currently working at Sheffield University. She has her undergrad degree in maths and statistics from the University of York, and at that time became interested in data analysis and machine learning. And from her undergrad degree, then went on to a PhD at the University of York in the biology department, applying her skills for biological research questions, where she did combine cell biology, machine learning, and technology developments in imaging for her work. And since I think last year, Laura has been a postdoctoral research in Sheffield, working on looking at DNA structure and DNA protein interactions. But I think today she will be telling us mostly about her PhD work on cell phenotyping.
Thank you very much for inviting me to give a talk today. I'm usually a very frequent attendee of the virtual pub, so it's really nice to be on this side of it. And what I wanted to talk about today is how you can make phenomenal insights using your single cell phenomics data. So I couldn't miss out on the opportunity to include a pun there. But what I'd like to do is raise awareness of time-lapse microscopy and its advantages over static time point microscopy, and also to discuss our analysis toolkit for time-lapse imaging, which is called Selfy. So I will begin with a little bit of background to the project. So I think one thing that we can all agree on is that our cells are very versatile. So they're able to adapt to their surroundings, they can respond to environmental cues, they can communicate with one another and coordinate their actions to achieve certain goals. But they're also able to possess this remarkable ability to resist treatment and to differentiate into different cell types so that they can achieve different functions. And we can gain a lot from techniques from genomics, transcriptomics, and proteomics to characterize these cellular processes in detail. But another thing that we can use to tell us about what our cells are actually doing is cell morphology and what cells actually look like. And because of that, if you're a trained biologist or if you spend a lot of time working with cells, you've managed to acquire this ability to look at cells just on the microscope and instantly know whether they're healthy, what cell type they are, if they've responded to a drug treatment that you've put on. And the reason for that is that there must be something about how a cell looks or metrics that our eyes are extracting from cells to tell us about what cell cycle or what cell state cells are at. But cellular processes are dynamic, and so a lot of microscopy tools that we currently use a static time point and may not be suitable to actually tell us a lot about these underlying processes. I think if we're going to characterize them in detail and fully understand what's happening to cells, we need to look at them over time rather than just taking static time points. So all of these cellular processes are happening and are possibly hidden within your microscopy images. But I think this is why we should use time-lapse imaging to get to the bottom of this, because otherwise these processes are just happening in the background and we've got the data on them, we just don't know how to extract that information. So one tool that we can use is microscopy, of course, and a commonly used microscopy technique is fluorescence imaging. But why that may not be so good for time-lapse imaging is because fluorescence heavily perturbs cell function, meaning that if we're extracting subtle changes in cell morphology, it could be that we're actually just looking at morphological descriptors that are changed due to fluorescent labels and heavy light exposure. And for that reason, for investigating such subtle differences and using time-lapse imaging, it would be better to use label-free imaging approaches so that we know that our cells are behaving as accurately and reliably as possible in similar conditions to how they would be in the body or in culture. And as we're able to image on a greater time scale due to advancements in label-free imaging techniques, so for example, we're able to image several experimental conditions in parallel for often weeks at a time, that means that we're generating a lot of data and a lot of images. And that means that actually the metrics that we can extract from that, such as shape, size, texture, and motility, we can extract those from every cell on every frame, in every imaged well. And actually, the volume of data that we're able to extract is enormous. So that leaves us with the question of how do we go about analysing that to get to the bottom of our biological research questions, or what we're trying to find out from our microscopy images. So that was the background and our motivation for the work that we've gone on to do. So we thought there's a need for label-free image analysis workflows. So they need to be high throughput, because we could be imaging several conditions in parallel. As I say, for weeks on end, that's a lot of data, and we need to be able to process that volume of data. But these analysis techniques need to incorporate temporal analysis. So it's not enough for us to say these cells are bigger, or they're this shape, or they've moved this much. We need to be able to say how those changes differ over time, and to compare those amongst different cell types. But they need to be user-friendly and reproducible. So it's not enough for me to have an analysis technique that works for my own cells on my computer and no one else's. It needs to be something that can be used by others and used by the community. It needs to be adaptable, so that it's not just working for one cell type or one experimental condition. And I think most importantly, we wanted the analysis techniques that we developed to be interpretable. So I know a lot of machine learning and deep learning can often be black box. You throw data in, you get results out, and you don't quite understand what's gone on in the middle. But what we wanted to do was develop an analysis method where you could understand what metrics were changing within your data and actually relate that back to your biological question, so that you can understand what's happening, and you can hopefully find out some super cool biology and science too. So I thought this was a great place to say thank you to the bioimage analysis community. It's a really collaborative community, and we've had some hugely great tools developed. It's a really active field. And I think what we wanted to do was develop a tool that doesn't compete with the current tools, but actually complements the data that you get out from those. So for example, recent advancements in segmentation and tracking are incredible. We're now able to segment and track with huge accuracy, really complex samples, highly dense cell samples, up to tissue, etc. And these are a really important starting point to be able to extract reliable phenotypic metrics and to go on and do something with that data. So what we wanted to do was develop a tool that uses segmentation and tracking as an input for what we want to do. And that means that hopefully, to be able to use our tool, which is Selfy, you can use any segmentation and tracking software that is suitable for your own application and use those as input for Selfy. So currently what those tools do is they facilitate the extraction of certain features from cells. So you can look at changes in shape and size, for example. Here's two examples looking at how circularity and area change over time. And what these tools facilitate is the ability to extract those metrics on each frame and plot them so that you can see visually what cells are doing over time. The problem is with that is that we have these time series, but we're not quite sure what to do with them. What metrics do we need to look at for single features, and how do we actually combine them? So how can we say one cell type changes in circularity and it also does this in area whilst doing this in volume, and kind of merging all of that together? And that was our starting point. So when we actually started this project, our first thought was, okay, we have all of these frames and we have all of these features happening on each frame. So we'll just do a big principal component analysis, treating each frame as though it's an individual, not considering that it's actually formed a time series. And we got this mess of a principal component plot where each color represents a different well on a multi-well plate from an experiment. And I think the first thing you can probably ask is what's going on in there. We've got lots of groups overlapping, and it's hard to tell if there's any variability between the groups. But what we wanted to do was kind of clean up this interpretation so that you're able to actually find patterns in groups and find what's different between them. So that was the motivation. So with that, I will introduce our solution, which is Selfie. So as I say, Selfie starts off hopefully with any cell type or imaging condition, any choice of pharmacology that you'd like to start with your experiment. And you then seed the cells and image them for a set amount of time. That can be whichever amount of time you need for your experiment. So I'll give a shout out to Phase Focus, who are giving the tech exchange after me. Throughout my experiments, I use the live site by Phase Focus, which is a really great system. It facilitates time-lapse imaging of multiple experimental conditions in parallel. And I'd also like to say that the University of York's Phase Focus system is part of Euro-BioImaging. So if you do want to use the live site there, please get in touch with Peter O'Toole, who's the head of imaging and cytometry in the Bioscience Technology Facility. And you'll be able to talk about either using the system or providing your own samples for them to image there. So that's just a little aside. But anyway, after time-lapse imaging, you can then go on to segment and track cells. So you can do this with any software that you choose. I stuck with the segmentation and tracking output from the Phase Focus, as that worked really well for my samples. But using those, you can then go on to use Selfie, which is where our own toolkit starts. And that facilitates the extraction of boundary coordinates so that you can go on to calculate a range of metrics for cells, including cell shape and size. We also use the interior pixels to calculate texture metrics, and we also calculate motility metrics as well. So that is for every cell on every frame. That can all be passed on, and we look at the time series of the cells and actually extract metrics from those too. So we're able to summarize cell time series through wavelet transforms and time series features as well. Using that, we have a segmentation error detection classifier. So I'm sure anyone who has performed segmentation before, this could be a very familiar site where you have either over-segmented cells or under-segmented cells. And what we found was that including those in our data sets really reduced the reliability and affected a lot of the biological conclusions that we were making. So we've developed a classifier that actually removes those for you so that you've got a cleaned up data set to carry on with. And then using your cleaned up data set, you can identify discriminatory metrics between your groups. So you can find which features will be relevant for your own analysis. And then there's also integrated machine learning capabilities. So you can go on to cluster cells, and you can also perform cell type classification if you want to do binary or even multi-class classification. So I will go on to explain each of these stages in detail, but that is just a little summary of what the Selfie toolkit actually aims to do. So in terms of quantifying cellular phenotypes, Selfie has a range of inbuilt features that you can calculate for each cell on each frame. These include a range of shape and size metrics. So I've included a lot of the mathsy formulas behind it just to show you that this is all actually done internally to Selfie. So all you need to do is plug your cells in and it'll calculate these for you. I think what's nice about Selfie is that these features are included in Selfie, but you can also include your own features. So for example, with the phase focus system, you also have the advantage of using the phase information to calculate mass and volume. So if you include those in your feature table, the rest of the Selfie toolkit will include those too. So perhaps if your own microscope has certain features that you're really interested in continuing analyzing, they'll be included here as well. So we also have motility metrics. And something to highlight here is, for example, we have displacement and track length. We also have displacement as a ratio of track length. So that's able to discriminate between cell types, perhaps that they have a large track length because they're moving far, but they're actually not moving too far from their starting position. So they're moving a lot, but only staying local to their central position. And we found that that was really useful in discriminating between cells that had a large track length because they were actually traveling or migrating far from their starting position. And then we also have texture analysis. So we use the interior pixels of cells to be able to look at the distribution of the pixel intensities within them. So the first set of texture metrics that we have actually perform wavelet transformations on the cells, and we calculate grey level co-occurrence matrices from these. And what we found that really useful for is being able to detect the cells that have these pixelated high intensities of clusters in the middle that you can see in that original image, and separating those from cells that perhaps are more uniform within their cell interior. We've also got a similar metric to that, which looks at pixel intensities and compares them to a spatial point process. Again, able to detect clustering of pixel intensities on different percentiles of intensity value. And what we found that really useful for is that, for example, with phase images, you have the pixel intensities relate to mass. And so this actually tells you something about the subcellular region within your cells. And it could be that you could use this for fluorescence images if you expect that the distribution of fluorescence might not be uniform or maybe uniform within your cell interior, but that will tell you something about those as well. So that's what you get from each cell on each frame. Now this is where we handle cell time series. So as I say before, it's not enough to just handle each frame separately. You have to handle them as a time series so that you can explore what cells are doing over time. So here's an example of a sphericity time series for a cell. And you can see that the cell has high sphericity in the top points and low sphericity and therefore long elongation in the bottom 2 points that I've highlighted there. And what's important is that each cell has a time series for every feature. So from Selfy alone, we can calculate 74 features, and each of those 74 features has a time series. So that is a lot of data. And time series can also be different sizes dependent on how long a cell is in a time-lapse form. And so it's important that we're able to compare these against each other despite them being different sizes. So what we do is we extract features from the time series. So for example, we were inspired by elevation profiles from walking guides, and we calculate total ascent, total descent, and maximum value from these time series. As well as that, we use wavelet transforms. So we don't just look at the original time series, we also decompose this into a series of wavelet transforms. So we look at their approximations and their details. And the reason for doing this is that we can reduce a lot of the noise from the original time series and be able to actually see patterns that maybe we missed from the original. So if you look at the level 3 approximation, you're able to see clearly that there's a decrease in volume, possibly a sharp decrease at the point between 15 and 30. And that wasn't as clear in the original time series. And so here we're able to actually look at changes in cell behavior at different temporal scales in comparison to what we were looking at the original time series. And importantly, using all of these time series features extracted from our phenotypic metrics, that equates to over 1,100 metrics per tracked cell. So there's an awful lot of information there about individual cells that you can then go on to use to answer your biological questions. So what can you do with those 1,100 metrics? As I said, the first thing that you can do is automate and remove segmentation errors. So segmentation errors really interrupt cell time series. On the left of those plots, you can see the volume and displacement for a cell that is correctly segmented. And on the right, you can see the same, but for an erroneously segmented cell. You can see that the time series look hugely different, and that's just based on the fact that there were segmentation errors within a cell time series. So we really want to remove those data points from our data set. So we've trained a series of decision trees to predict whether they think a cell is a segmentation error or whether it's a correct instance of segmentation. And based on majority vote, we get a final classification of whether it's correct or segmentation error. And here are some examples of our model performance. So the ones on the left are predicted correct segmentation. As you can see, they are correctly segmented cells. The ones on the right are those that were predicted to be segmentation error, and they are instances where multiple cells have been segmented together. So that's just from our classifier learning the time series behavior of a segmentation error and comparing that with the segmentation, the correctly segmented time series. So what else can you do? You can also identify discriminatory variables. So often you'll have two different populations. You want to know what's different between them. And we use a metric called separation scores. So what that looks for is you want a metric that has a high between groups variance. So if you look at the between groups variance there, you want the ones that are high because that means it's separating out your two populations well. But you want a metric, with within groups variance that is low because you want all of the cells within that population to act similar for a certain metric. So say it is curvature. If curvature has a high separation score, then your cell type A and B will have different curvature, but all of your cells in cell type A will behave really similarly for curvature. So that's how we measure how good a feature is. And we use this as a way of reducing the dimensionality of your data set so that you only move forward with the features that are actually of importance for your biological question and remove any that are redundant variables. And then, as I said earlier, Selfie has integrated machine learning facilities. So it's got ensemble cell type classification, makes use of several machine learning classifiers to predict whether a cell is cell type A or B. And then the final prediction is based on majority vote. And also we have a range of visual facilities for your cell type. So we've got t-SNE, we've also got PCA and UMAP. So all you have to do is plug your data sets in and you can produce these plots and edit the parameters as you see fit. As well as that, there is cluster analysis capabilities. So that's hierarchical clustering and k-means. And we do have a method included, which is the within sum of squares. So you can get an idea of how many clusters you can expect your data set to have. And then you can visually inspect the plots to see which clusters suit your data set best. So the question is, how can you actually use Selfie? So Selfie is currently an R package. It comes with a lot of documentation to help you get up and running with the functions that we've got. It's easy to install, and there's customizable parameters so that you're able to edit our own workflow as you see fit for your biological questions. We have got an in-depth user guide, which does have a worked example provided. As well as that, there's also a Selfie GUI, which I prefer for easy visualization of your data. You can plug in your data sets, you get all of this analysis, so you can look at the features of importance, you can do your cell type classification, your cluster analysis. And the nice part about this is there's very minimal coding required, and there is a demonstration video to help you get to grips with that as well. So just to point you in the right direction of all of that useful information and resources for you, in the main Selfie publication, we have a link to our GitHub repository. There's a lot of useful documentation on there to help you get started with Selfie. You can find our Selfie user guide within the supplementary information on the Selfie manuscript. As well as that, there's also the demonstration video, which is in the data availability section of the Selfie paper. And that includes a worked example and my little face in the corner explaining how to do everything. So that could be really useful to watch as well. Okay, so now I'll go on to talk about a few different case studies of the interesting biological applications we've been able to look at with Selfie, and hopefully give you some hinters on how you can make use of Selfie within your own work. So the first one is characterizing treatment response. So the motivation behind this is that it's known that for a culture of cancer cells that are treated with chemotherapy, a subset of those are successfully treated, and there's always a subset that acquire resistance or have intrinsic resistance, and that population go on to proliferate further, and often they cause cancer progression, usually in a worse state than the original cancer. So what we wanted to know was, can we use Selfie to identify the phenotypic markers of resistance and actually understand which cell types acquire resistance and which ones are successfully treated? So the key questions were, can we characterize phenotypic changes in untreated and treated cell behavior? Can we classify it? Because if we can, then we can use predictive models to say whether an unknown cell has successfully responded to drug or not. And then can we use that as a way to identify chemo-resistant cells? So using Selfie's feature selection, we wanted to see which phenotypic metrics differ between untreated and treated cells. And doing that, we found that untreated and treated cells differed mainly in texture, shape, and size. So the reason for that is that we treated with a drug called docetaxel, which is a microtubule stabilizing chemotherapy drug. And when we treat the cells, they're unable to divide, so they grow large in shape and size. And the texture metrics that we were pulling out as being significantly different were because the inability to divide means that the cells become multinucleated. So the texture differences we're seeing are actually pixel intensities within the cell as a result of multiple nuclei within there. So the features that we were extracting was telling us something about the drug mechanism of action, which was really interesting. And then using those features that were identified as being important, we were able to nicely separate out the untreated and treated populations. And we were able to generate these phenotypic profiles of what it looks like to be an untreated cell and what it looks like to be a treated cell. So the untreated population are much more motile in each feature, and they fluctuate a lot more in shape, size, motility, and texture. Treated cells are usually a lot more stable across a range of time series. So to check that that wasn't just an artifact of our cell type and our drug, and to check that this would actually adapt to different cell types and drugs, we tried another cell type and another drug called doxorubicin that has a different mechanism of action. So for the different cell type treated with docetaxel, we got similar texture metrics out. And the reason for that is that we were able to characterize the docetaxel effect for a different cell type, but we were actually seeing the same multinucleation and shape and size difference as we were seeing for the previous cell type. Interestingly, for doxorubicin, the main mechanism of action actually is to inhibit cell migration. So you can see a lot of lilac points at the top there. That's because movement metrics were affected. And so we were able to say which features were different between different drugs, and that was telling us about drug mechanism of action. So that has quite a nice application for drug screening, perhaps if you go into the experiment not knowing which phenotypic metrics will be differed by your drug. And this gives you quite a nice way of saying what exactly has happened to your cells upon drug treatment. And as before, we were able to characterize these populations and separate them out into treated and untreated, where more overlap shows cells that have been treated but are still behaving as though they haven't and are therefore suggestive of drug resistance. So now we wanted to predict, has a cell responded to drug or not, again using this cell type classification. So interestingly, what we found was across all of the experimental conditions, there's always more treated cells classified as untreated. So these cells have received drug, but they don't appear to look like a treated cell based on their phenotypic metrics. And we think the reason for that is something called hidden stratification. So that is when machine learning models underperform on important but unrecognized subgroups of the data. So what that means is that in our treated population, we will have subpopulations of successfully treated cells, that have responded to drug, and therefore look how we would expect a treated cell to look. But we'll also have a subpopulation of cells that are resistant, and therefore they will be classified as untreated because they look as though they're untreated. So to test this hypothesis further, we went on to investigate cells that were treated but looked like an untreated cell. And that's these cells. And you can see that their phenotypic profiles do look really similar to those of the untreated population. We then treated with increasing concentrations of drug, expecting that we may see more resistance at the lower concentrations where cells haven't responded as well to higher concentrations of drugs. And that's exactly what we saw. So we saw that as we increased drug concentration, we had more cells classified as treated. And therefore, we were able to quantify on a population level how much or how little a population had responded to the drug. And that's just using Selfie's cell type classification models. So what we did then was within the treated cell population, we wanted to see, can we identify heterogeneous phenotypes? And we were able to, and you can see that there's a wide variety of phenotypes just within one treated cell population. Nicely, there is linked to cell cycle stage. So we use mean volume as a measure of cell cycle stage. And we found that, for example, the cells on the far left in that cyan colour, they're cell debris, where their mean volume is heavily decreased as they've died. And we also saw that the pink and the blue populations, which are those really large cells in 3 and 5, they've escaped the cell cycle and continue to increase in mean volume. So we're able to identify heterogeneous phenotypes, but also link those to the underlying biology of the cells. And even for populations that looked quite similar by eye, we were able to use Selfie's feature selection to identify which metrics actually differed between those populations, even if those differences were really subtle, we couldn't see by eye. So going on to case study 2, we thought we want to characterize interactions between cancer cells and cancer-associated fibroblasts. So all of the work that I've presented till now has just been on single cell types or monocultures of cells, whereas in reality, the tumor microenvironment is actually extremely heterogeneous, and you often get several cell types communicating with one another and changing the behavior of cell types around them. So the cells that we wanted to focus on were cancer-associated fibroblasts. They're known to encourage cancer cell growth, survival, progression. They promote epithelial to mesenchymal transition. They induce cancer stem cells, and they also induce chemotherapy resistance. So we thought all of those sound as though they will induce morphological differences in cells that hopefully Selfie is able to detect. And all of this is mediated by direct contact of cells, as well as growth factors and cytokines. So it seemed like a perfect application for Selfie. So the first thing that we found was that CAF-conditioned medium actually changed breast cancer cell morphology. So you can see it's a lot easier to see by eye, I think, on the bottom row for MCF7s. The control samples act like epithelial cells, as you'd expect. They stay quite close to one another, and there's not much movement, whereas you can see an increase in motility as cells begin to move away from one another once they've been treated with CAF-conditioned medium. So that tells us that there's something within the conditioned medium that is actually making cells respond differently. And we're actually able to quantify those differences using Selfie. So when we did that, for both the cell types that we tested, we were able to see differences in morphology across all of our feature types. So texture, shape, size, movement, and density. And some of those differences were actually quite large. So some of the ones that are interesting to look at are things like sphericity. So cells became a lot less spherical when they'd been treated with CAF-conditioned medium. They became a lot more elongated in shape, which is a phenotype associated with mesenchymal cells, which is a more aggressive type of cancer cell. And we also noticed that they fluctuated a lot more in their adherence as they were able to contract and protrude, as they were able to move around more, so increased migration too. So all of these metrics were associated with an increase in the aggression of breast cancer cell phenotype. So that was just induced by treating with CAF conditioned medium. So the reason that people usually treat with conditioned medium rather than actually co-culturing cells is because it can be really hard to separate two cell types out after imaging. But we thought that this would be quite a nice challenge for Selfy to see if, just based on phenotypic metrics alone, we could separate out cancer cells and fibroblasts after imaging. And nicely, we were able to do that. So we were able to say which phenotypic metrics differed between cancer cells and fibroblasts. And then we were able to do essentially a label-free cell sorting. So the fluorescence that you see here was only used as ground truth just to check our accuracy. But you can see from the test set results that we were able to nicely separate out cancer cells and fibroblasts. And why that's really useful is then we can go on to isolate the population of cancer cells and actually investigate what phenotypic differences were induced based on co-culturing alongside CAFs. So to test this further, we went on to treat with increasing concentrations of CAFs with the hope that if it is CAFs that are mediating these phenotypic changes, we should see a greater amount of phenotypic changes when cells have been treated with a higher concentration of CAFs. And that is exactly what we saw. So you can see here from the proportion of CAFs and from the PCA plots that there is a phenotypic shift induced by treating with CAFs, which you can see by the ellipses moving from the control point. There's actually less overlap between the ellipses for the control set and the 75% proportion of CAFs. And the reason for that is because the higher number of CAFs that you co-culture the breast cancer cells with, the more phenotypic differences you can see in the cancer cells. So we're able to quantify how different cells are behaving, as well as see what's impacting that. And that's further supported by looking at single feature plots. So you can see on each of these plots, there's either a gradual increase or a gradual decrease across a range of features, that is based on the proportion of CAFs that we've added to the co-culture. So we can see that these phenotypic differences are mediated by just co-culturing with CAFs. So we wanted to check, can we actually correlate our findings from imaging with different technologies to see if we can gain more information from what we found with imaging. So we performed a multiplex immunoassay to check what was actually in the CAF conditioned media that could be inducing what we think is evidence of epithelial to mesenchymal transition within the cancer cells. And we did find a cluster of analytes within the media that are actually described in the literature to induce an aggressive phenotype. So that sheds light on what could actually be happening, or the environment that the cells are in that could be inducing these phenotypic changes. And we also found a cluster of analytes where the production was inhibited following treatment with CAF conditioned media. So that means that not only is it the CAFs that are retaliating to the cancer cells are actually speaking back to the CAFs and changing their own production based on signals that they're getting from the CAFs. So we're able to see evidence of a crosstalk between CAFs and cancer cells. And again, another technique that we've looked at correlating our imaging data with is RNA sequencing. So we wanted to see, are there any differences in gene expression when cells have been cultured in CAF conditioned medium? And particularly for our MCF7 cells, we have found some genes that are differentially expressed after treatment with CAF conditioned medium. And again, in the literature, we found that these are linked to increased tumorigenesis and aggression in cancer cells. So it looks like there's a neat way of tying it together that the conditioned medium induces phenotypic changes that actually links with the underlying biology of the cell. So that's just a nice idea of how you compare phenotypic information with already traditional techniques. And then my final case study is how we can use Selfie to look at multi-class classification. So everything I've shown so far has been binary classification, but as we know, you can have multiple cell types in one culture in reality in your biological experiments. So what we did was we imaged several breast cell lines, 2 examples of triple negative breast cancer. We have a luminal A breast cancer, and we also have normal epithelial breast cells and fibroblast cells too. And the reason that we image these is because we know that they have different genotype profiles, but we wanted to see if we could separate these cell types out based on their phenotype rather than any other experimental tests that we could do to check differences between them. So when we did this, we looked at just the pairwise separation scores, first of all, using Selfie. And you can see that binary classification wise, you're able to get really nice separation between each cell type there. And then pairing that all together for multi-class classification, we found that we're able to separate out populations of different breast subtypes. And what that tells us is that we could begin to use this as a sort of phenotypic atlas to say, here's an unknown cell. What type of cell is that based on its phenotype alone without any genetic information? And interestingly, although you can see the colours are reflective of cell line, there's also 3 main clusters that you can see there too. And they're separating cells out into mesenchymal cells, which is your turquoise and blue there. You've also got epithelial cells, which are your green, yellow, and magenta. And then you've also got fibroblast cells, which are in red. So it's actually showing that Selfie can separate out distinct cell lines, but can also separate out into mesenchymal properties, epithelial, and fibroblastic too. So finally, we just wanted to check, can we use this in actual primary cells rather than just looking at cell lines? So we imaged some healthy primary breast cells, and we used our classification model to see what sort of cell type did they look most like. And we found the highest proportion of those looked like MCF10As, which are our cell line, which are representative of healthy primary breast cells. So that tells us that most of our cells are being identified as healthy, and shows that there could be something in predicting cell type based on phenotypic metrics alone. What you'll see here is that there are still percentages that were incorrectly identified as some of the cancerous cell types. The reason for that is that a lot of the cells were incorrectly segmented and tracked, as you can see from the really high density of cells within the image. It presents a really challenging problem to be able to segment and track the cells accurately, which is quite a nice segue into the last bit of the talk, which is, so what's next for Selfie? So I've shown you all the cool things that you can do with Selfie so far, but what I'd like to introduce is that we're now planning to work on Selfie 2, and I will tell you a little bit more about our plans for that on the next slide. So what have we got planned for Selfie 2? So we're planning to extend to additional imaging modalities. So the work I've shown you here has been looking at tachographic images, but we're looking to extend to bright field and fluorescence. We've got a contrast phase imaging experiment going on next week, so hopefully we'll be able to include that to the list as well. And we're always looking for different imaging modalities and different samples, so we want Selfie to be developed in line with experimental needs. So if you have any samples that you'd really like to be able to use Selfie on and analyze those samples, then please do get in touch and let us know. Selfie 2 will also have new analysis capabilities. So that last example I showed you there is an example of a pretty complex biological sample to analyze. So one thing that we want to incorporate is whole frame analysis, which will be able to handle those situations where segmentation and tracking isn't as possible as for easier samples. We also want to incorporate tracklets, which will be able to fragment whole cell time series and hopefully shed light on timescales within your data set where interesting behavior is happening, but you're currently missing it when comparing just whole cell time series. And we also want to enable identification of early predictors so that you can say from your time series that if a particular cell acts this way at the start of the time series, it's likely that they will perhaps acquire drug resistance, or they may go on to differentiate into a different cell. So we want to improve the types of biological analysis that you can get from your data sets. And a really exciting thing that's coming with Selfy too is a cell profiler and Selfy collaboration. So what we're aiming to do here is to integrate Selfy into Cell Profiler. So you'll be able to use Cell Profiler workflows for your segmentation and tracking, and also all the neat feature extraction that they've got there. You'll then be able to go on to calculate all of the time series information from Selfy and perform all of the analysis from within Cell Profiler and Cell Profiler Analyst. And part of this collaboration means that the current R package for Selfy will be rewritten in Python. So hopefully for all the Python fans out there, that's really good news for you guys. And you'll be able to use Selfy and use some of its modules within your own analysis workflows as well. So that's our plans. So that's all super exciting. So with that, I will summarize. So Selfy is cell phenotyping made easy. The input is time-lapse images and tracked segmentation, which can be from any of your favorite segmentation and tracking software. And then you get all of these cool outputs. So over 1,000 phenotypic metrics, removal of segmentation errors. You can identify features of interest, cell type classification, and cluster analysis. And this should work for all cell types and all live cell imaging applications. So hopefully you can find use for it within your own work. And with that, I would like to acknowledge everybody who's been part of this project. We've had a really great team, a great interdisciplinary team working on this, which has been brilliant. And of course, if you're interested in using Selfy or have any ideas for things you'd like to see from Selfy too, then please do feel free to email and get in touch with us, because we'd love to develop it in line with user needs. So thank you very much for listening.